In today's lecture, we're going to begin the first of a few multi-part series on the nervous system. Today, we're going to specifically focus on nervous tissue and then organization of that tissue. As always, you want to make sure that you're able to execute these different learning objectives before you proceed to the next lecture. Okay, so let's go ahead and get started here. So the first question we want to ask is, what are the three overlapping functions of the nervous system? Sort of a big picture question. Let's go ahead and take a look at this. First, we have something called sensory input, right? Your body senses something from the environment. Whenever it's coming in from the environment, we call it afferent. Underneath the letter A, put a little line there, right? Afferent, coming in. The next thing is something called integration. And integration is sort of a processing or interpretation of the sensory input. And this happens inside the brain itself. Third, we have a motor output. Once a decision is made, then your body acts in a certain way. And whenever you have this motor output, the movement's coming out, we use the word efferent with the letter E to describe this. One thing you want to note too is each of these lines here, this blue line, this gray line, and this red line, can be indicative of neurons that are helping to execute this function. Okay, so what are the main divisions of this of this nervous system? Well, if we look at the human body, we'll see that some are here in beige, right? Some are here in yellow. So what do these colors represent? Well, the beige represents the brain and the spinal cord. This is something called the central nervous system, or the CNS. Then we have the nerves and something called ganglia. So what ganglia are basically areas where the cell bodies of neurons cluster, right? Nerves are basically, bunches of neurons that extend from the brain and the spinal cord. And these two things together are called the PNS, or the peripheral nervous system. Together, these make the two main divisions of the nervous system. If we look at a little chart here, we can classify different types of basically sensory and motor fibers in the peripheral nervous system. So let's go ahead and look at this. So if we look at the sensory components, we can classify in two different areas here. We have somatic sensory, which are things like touch, pressure, temperature, proprioception of the skin. Proprioception means, it's a fancy way of saying your ability to basically tell where your body is in three-dimensional space. So like a gymnast, for example, would have very good proprioception, very good awareness of where their body is in space as they're on the balance beam, something like that. Specialized versions of this type of somatic sensory are things like hearing, equilibrium, vision. When we contrast it, you'll get the difference here. If we have visceral sensory, these are things that happen sort of inside your body. So whenever we think of the word visceral, we think of like inside. So like stretch or pain or temperature, the feeling of being nauseous or having hunger pains. Those are all examples of general visceral sensory. Special examples are taste and smell. Again, things that are happening sort of like inside your body. The stimulus is from inside your body. Okay, so those are the two types of sensory components. If we look at the motor components, we would say that we have somatic motor, and this is sort of, again, soma means body in Greek, and here we're just saying literally your muscles, right? Skeletal muscles moving. That would be sort of somatic, emphasis on the skeletal. Then we have visceral motor, and this is something that is part of the autonomic nervous system. And what this is for example, when you eat and then your body is releasing digestive juices in your stomach, like hydrochloric acid or pepsin, things of that regard, that would be visceral motor, right? So motor that you cannot really control, and that's what this word here means. Okay, so what are the main characteristics of the nervous system, of nervous tissue? There's a few things we want to note here. The cells are very densely packed together and intertwined, right? And there's two main types of cells. So there's neurons. These are the cells that actually do the transport of electrical signal, right? So those are the main players. But there's a lot of support cells as well, things called neuroglia. Those are support cells. And they're non-excitable, but they wrap around neurons, and they have many, many different functions that we'll talk about throughout this lecture. A little bit later on, we'll talk about the different supporting cells in the central nervous system as well as those in the peripheral nervous system. Another thing we want to note is that neurons are the basic structural unit of the nervous system, right? They conduct those electrical impulses along their plasma membranes. Something we call the nerve impulse, or it's an action potential that travels down the membrane. We've talked about this before. A word we often associate with the beginning of this cascade of electrical impulse is something called depolarization. Okay, and then finally there's three main special characteristics we would use to classify neurons. So the first one is longevity. In other words, neurons can live and function for a very long time, throughout a lifetime. And the second one sort of tells you why that better be true. Well, they can't really divide, right? And so if they can't divide, we can't really produce more of them, then they better last a long time, otherwise we're in trouble. Now, that's a little bit debatable nowadays. We are discovering there are some neural stem cells, right? There's sort of an exception to that, where maybe we do have some division of neurons, after birth, although, that's not typically something we think of. So we're learning more and more there is some credence to that. And then finally, the third major note or characteristic is that they have a high metabolic rate. And this makes sense. If your neurons go without oxygen for about 5 minutes, they will die. And that's why if someone's having a stroke, it's very important that they actually seek medical attention very quickly to restore that blood flow to the brain. Otherwise, within 5 minutes, they're going to start losing neurons, if not sooner. Okay, so what's the anatomy of a typical motor neuron? There's a lot of different types of neurons we're going to talk about, but a typical motor one, what's the anatomy of it? Well, there's a few different parts we really want to note here, right? So the first part is something called the cell body. And the cell body is indicated right here by these circles I'm drawing, right? So these are sort of the cell bodies. And the cell body basically contains the nucleus, right? Houses the DNA, the nucleolus. Really, it does all the main functions of what we think about when we think about a cell, right? It has the mitochondria in there, etc. Okay, so nothing too special about that in a sense, right? Although there are a lot of mitochondria in those neural cell bodies. The second thing we want to talk about is neurons have these things called dendrites that are very often, in this case specifically, reaching off the cell body. So you'll see them here. And these dendrites, what they are is they are actually the receptive regions, or the things that are going to receive the sensory input from the external environment. So the input is received usually via some chemical, something called a neurotransmitter, I would write down, right? And then the dendrites receive that info, and then basically that info is somehow transferred through this thing called an axon, specifically at the axon hillock. And at this point here, the axon, what happens is that chemical signal, right? So emphasis on the chemical, received via neurotransmitter, is translated or transferred into an electrical signal. So at the beginning of the axon, we have an electrical signal here happening right at the axon hillock, this region right here, and then going down the axon, right? So electrical signals. So transferring chemical to electrical. It's very important to note that. It'll travel through the axon hillock very quickly, in many cases extremely quickly, because you have these things called Schwann cells that create something called myelin, right? A myelin sheath around the axon, and that myelin sheath sort of acts as like a conductive insulator, like sort of two opposite words there. But basically what they do is they coat the axon and they allow the electrical impulse to travel very, very quickly. So it's not dissipating into the environment. That's where I got the insulator part, right? But rather it's the impulse is traveling right along the axon very, very quickly. Okay. Then finally, the third main part of the neuron we want to think of is something called the terminal bouton at the end. And what happens is that electrical signal that you'll see right here where I'm drawing this asterisk will again reach down these branches and be converted to a chemical signal and then released into something called a synapse, which is the space between this neuron that you see right here and the next neuron that's waiting for it. The next neuron that's waiting will have its cell body over here, its dendrites reaching out, and it's ready to grab that chemical signal you see right here, transfer it down its axon, and then release it via its terminal boutons again. So it's a really neat cascade you see there. So it's a nice little diagram of a neuron. You want to know all these different components of it. And then here obviously is a nice picture where that's what a real neuron looks like. Again, the real image is never exactly the same as our cartoon. Okay, when you think of a neuron transferring a signal, I think a good way to think about it is old broadcast TV. So receiving basically a TV signal via an antenna on top of your house, transferring it down a wire, right? So the antenna would be the dendrite, the wire would be the axon, and then coming into the TV and releasing it as a chemical signal or a TV picture. So the TV would be the terminal bouton. Sort of a neat way to think of it. A few other notes to note here. I'm sort of blocking them with my little writing here. I apologize, but on your PowerPoint you'll see it. Most neural cell bodies are located within the CNS, right, the central nervous system. When you have neural cell bodies in the peripheral nervous system I mentioned on the previous slide, they're called ganglia. Then one more thing to mention, if you have a very long axon, it's called a nerve fiber. Okay, let's continue here. So how is an action potential turned into a chemical signal? Well, basically the reasons I just mentioned, right? And this is sort of a nice picture showing you that. So in between these two neurons, we have a space. So if I looked at this picture right here and I blew it up, I would see what I'm seeing on the right here. So it's hard to see the space over here, right? But you could see that space here. And so really this is showing what I showed you on the previous slide, just blowing it up a little bit. It's a nice diagram to look at. So the signal would be coming, electrical signal down this neuron here, released as a chemical signal, right, in this circle right here. And then if we blow that circle up, right? We're sort of looking down here now. Here's the chemical signal being released, all these neurotransmitters, and they're being received, right, by this dendrite here, which is the dendrite right here of this next neuron waiting to receive that signal. So that's how an action potential is turned into a chemical signal, transferred from one neuron to the next. Again, you want to notice that what is this space between neurons called? It's either called a synapse or a synaptic cleft. Either one is a correct way to say it. They're synonyms. Okay, we can also classify neurons based on their structure, right? So different structural classifications. So they can either be multipolar, they can be bipolar, or they can be unipolar. And all these things refer to are the number of projections that are coming off the cell body. So we've got to look at it very carefully here. So if we have multipolar, we're saying here's the cell body in the first case, and you have all these different dendrites coming off, right? If you have all these dendrites coming off, that's why it's multipolar. You have many, many projections, right, or things, or dendrites in this case, projecting off the cell body. If it's bipolar in the second case, you could see that here's our cell body, and we have one projection here and one projection here, right? So two, so it's bipolar. And then unipolar, you have your cell body here, and you can see you have one tiny little projection coming off that cell body, and that's it. And so that tiny single projection there is why it's unipolar, or one. If we look at that, there's different functional regions associated with these different types of neurons. So you have something called the receptive region that you'll see here, something called the conductive region, something called the secretory region. And all that's referring to is, where it's receiving the stimulus, right, via the dendrites, where it's conducting down the axon, and then when it's secreting via the terminal batons. And it's showing you how these different regions differ depending on which type of neuron you have, whether it's multi, bi, or unipolar. And then these regions, these axons, these different types are found in different situations or different areas of the body. By far the most common is the multipolar, and you can see that sort of down here, right? It's the most common by far. You see it, throughout the body. The bipolar is very rare, though you do see it in the eyes or the ears, and we're going to see that later on in lab, actually. And then the final one is the unipolar, which is found mainly in the peripheral nervous system, specifically in the sensory ganglia of the cranial nerves. Okay, so all neurons are associated with what type of non-nervous supporting cells? What are they called? Well, I mentioned before they're called neuroglia, right? There's 6 types of neuroglia that you want to know. 4 of them are in the central nervous system, 2 are in the peripheral nervous system, and they provide support functions for the different neurons. And we're going to talk about this in the next 2 slides. You want to make sure you know these support functions, at least be able to name a couple things about them. They cover non-synaptic regions of the neurons. So the neurons are not just interacting with, solutions surrounding them, but really they're interacting with these support cells. Okay, so what are the neuroglia in the central nervous system? Well, if we talk about those, basically we want to see that they outnumber the neurons 10 to 1. So there's a lot more support cells than actual neurons, which is sort of interesting. Make up a huge amount of the mass of the brain, right? And they can divide throughout your life. So let's look at those here. So let's focus on the central nervous system first and the 4 types of neuroglia we find there. So the first type are called astrocytes. You see an astrocyte right here. If we talk about the functions of them, what are they? Well, not totally understood, but we know a little bit about them, right? So they can regulate neurotransmitter levels. They can signal to increase blood flow, right? They can control the ionic environment. When we look at these here, if they can regulate neurotransmitter level, these astrocytes might be a very good therapeutic target if we're looking for certain types of medications that would affect neurotransmitter release in someone's brain. So if someone's suffering from depression or anxiety, there's many ways to target that or help improve those situations. But targeting the astrocytes is something we realize nowadays that might be a new type of therapy for that, because we can regulate the neurotransmitter levels in the brain that affect those different mental health type issues, anxiety, depression, and there's others as well. Okay, the second type of neuroglial cell in the central nervous system is something called a microglial cell. And these are defensive cells in the central nervous system, right? So they're very small and at least abundant. But basically what they do is they're sort of like the macrophages of the central nervous system, right? So we have something that we call the blood-brain barrier. So it really helps prevent a lot of microorganisms from entering into the brain. But if they do happen, you have these cells to help combat those. So someone doesn't develop an infection in their brain, which would be a very serious condition. What do? They engulf these microorganisms, right, in dead neurons, and they dispose of them. They're derived from blood cells called monocytes. So really they are the macrophages of the central nervous system. Okay, The next thing we want to talk about here is something called ependymal cells. And really what these are is they line the central cavity of the spinal cord and the brain, and they have very, very tight junctions. So if you look at them, you'll see that there's not much space between them. I can barely draw my pen between them. In fact, I probably can't. I'm sort of forcing it in there. And that's important, right? So it's a very solid barrier, between these, cerebrospinal fluid and the external environment. They have cilia, right? So they can push substances around to help circulate the cerebrospinal fluid. Whenever you see cilia, right, you want to think transporting substances, moving them around. And then finally, the fourth type of neuroglial cell in the central nervous system is something called an oligodendrocyte. And really what these do is they help produce myelin sheath, right, around the axons of neurons. But what they do is they do it in the central nervous system, right? So there's two types of cells that do this. The oligodendrocytes do it in the central nervous system, whereas something else called a Schwann cell, you'll see it on the next slide, does it in the peripheral nervous system. Okay, so let's go ahead and talk about those other things now. Let's talk about the two neuroglial cells in the peripheral nervous system. Well, there's two here, right? There's satellite cells that you see right here, and there's Schwann cells that you see right here. The satellite cells surround the cell bodies within the ganglia, really sort of a source of protection, right? That's what they do. The Schwann cells are something we think of a lot more because what they do is they surround the axons in the peripheral nervous system, and they help form those myelin sheaths that we were talking about. And they help increase the speed or the conductivity of the electrical signal along the axon. So they're very important. A lot of times if people have trouble with their myelin sheaths, it can result in situations where they develop things like multiple sclerosis or MS. Okay, so what is the gross anatomy of a nerve? Now that we talked about this sort of microanatomy part, let's look at the gross anatomy, the bigger picture where you can see it with your naked eye, or at least some of it with your naked eye. So let's go ahead and look at this. We have some terms that are easily confused. We've got to make sure we pin down here. So a neuron is a nerve cell, right? When we say that, so it's a physical cell. If we say nerve fiber, it's a neuron that has a very, very long axon. And then if we say the word nerve, really that's a collection of neurons, or specifically axons, right, in the peripheral nervous system. So it's a collection of them. If a Q-tip was a neuron, a nerve would be like taking a bunch of Q-tips in your hand and holding them together. Okay, and this is what this picture is trying to simulate here. So if we have an axon up here from a single neuron, right, so this is our Q-tip, if you will, and has that myelin sheath along it, right? You put that Q-tip is surrounded by a tissue called endoneurium, right? Endoneurium. You put a bunch of those together, you get something called a fascicle, right? That's surrounded by a tissue called perineurium. And then if you put a bunch of fascicles together, that's surrounded by another sheath, another tissue called epineurium. If this looks familiar to you, I hope it is, and think about where you saw this before. Where did you see it before? If you're thinking in the muscle lecture, you're 100% right. These terms are almost identical to the muscle terms, except instead of having myo, right, we have endo, and then there's slight differences in the words from there, but very similar structure. The structure is nice because it allows passage of blood vessels, right, through those nerves. Okay, and these are some actual pictures here on the right that show you that. Okay, so how are the central and peripheral nervous systems structurally and functionally integrated? How do they work together? It's sort of a nice picture here. So you'll see the peripheral nervous system here, right? So you see nerves entering and leaving the spinal cord. You have the central nervous system that's composed of the brain and the spinal cord. Let's look at the central first, and then we'll go to the peripheral. So in the internal part of the central nervous system, you have this H-like structure of something called gray matter, right? And the gray matter really has a lot of things in it called interneurons, or the cell bodies of interneurons. So this is an example right here of an interneuron. It's a neuron that connects the neuron coming into the spinal cord with the neuron leaving the spinal cord. So the sensory input coming in with the motor the motor output leaving the spinal cord. And really what the interneuron does is it helps connect that in there. Sometimes you'll have another neuron branching to the brain if your brain has to register the signal and make a decision. Sometimes if it's a reflex, you might just have a situation that we see here. So that's sort of neat. Around the gray matter, which is in the center of the spinal cord in the H-like structure, right, we have something called white matter. So we have that here. And the white matter is where we have these very long neurons that have very long axons, right, that are called fiber tracts. And so we have those running through the white matter. Okay, at the very center of the spinal cord is something called the central cavity. And then if we look at the peripheral nervous system here on the right, we want to note a few different things here. So the sensory input, or the afferent input that we talked about before, right, is coming in along this track here, and it's coming in the back part of the spinal cord, or that would be the dorsal or the posterior part. So I would write dorsal or posterior back there, right? Sensory input always comes in there. Then it's integrated, right? And then what happens is you have the motor output, or the efferent output, leaving the front of the spinal cord. And the front of the spinal cord is called the anterior side, right? Or the ventral side. Okay, these are just some things that mention what I just talked about before, just to make sure you got these in your notes at the bottom here. Okay, I think I covered all that, but good. So that's how they're integrated, these two systems. It's really a highly organized system. You'll notice it's a sort of a interesting little pathway that we have there. Okay, let's look further into this, right? And so here you see sort of a diagram showing you almost the same thing as the previous slide, but sort of, putting some extra terms here, in terms of if you poked your skin with a pin, right, or a nail, you would feel that in the reception, right? So the dendrites there would receive that. It would go down the sensory neuron, go to the integration center, right, which is this interneuron, and then leave the motor neuron, which is efferent. So pretty much the same thing as the last slide, but just showing you how that works there. Let's add some terms in there, describe them a little bit, right? So we're putting some words with those terms. So I would read over everything on the red here. We describe this whole process as something called a reflex arc, right? But really I covered most of it on the last slide. Just a nice little review on this slide for you. Okay, so how do interneurons of the CNS, or the central nervous system, relay information to your brain? So this is going to show you that. So there's two different ways here. And so the first situation here is something, let's draw a line here. We just have a nice little dividing line right here, okay? So everything on the left is sort of one situation we're going to talk about, something called the spinal pathway. And then everything on the right is going to be something called the cortical pathway. It's a little slower, right? So in the spinal pathway, what happens is you're having something called a withdrawal reflex happening. And so really what this is it's an emergency situation. You touch a hot stove, or a hot match in this case, right? So really what's happening is that sensory input's coming in. There's an interneuron, and then sensory input's on the blue line, right? The interneuron there is in that dark blue sort of purple line. And then in the red line, you have the motor output, right? Boom, 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 it happens. In other words, you withdraw your finger from that heat, and this is the key point. I would write this down. You withdraw your finger from the heat, from that hot stove or that hot match, before your brain even knows it happened. Now, obviously, there is a relay to the brain. You can see there's no neural input into the brain here, but it's happening slightly after you actually conduct the act, which is really sort of a neat thing when you think about it. Okay, and that's important, right? It's a survival mechanism. Now, we contrast that to something called the cortical pathway, and that's everything on the right here. It's a bit slower here. So what's happening in this cortical pathway, if you read through it here, is you have something called parallel processing, right? So the sensory input comes in, and then what happens is that sensory input goes up to the brain, and then the brain goes back, makes a decision, right? Then sends that sensory input back down, and then it results in a motor output. And so the rest of this slide is showing that. So if you look on your PowerPoint, you'll see the inactive regions are the neurons that are not active at an exact moment are sort of, they're dull, they're sort of faded, right? Whereas the ones that are active are showing you how this actually happens. And so it's really sort of a neat thing here. And by the end, what you'll notice is that the motor response you do is voluntary. You're choosing to go, after you realize this happened, you're choosing to go to the sink, and you're cooling your hand off with some cold water after you burned it. So really, the one on the left is involuntary, the one on the right is voluntary. Okay, so what are some examples of disorders of the nervous system as we wind up this lecture here? Well, there's one I mentioned before, right? And it has to do with these myelin sheaths. And so here you see the axon running through the middle of this picture, right? And everything that's just outside of it right here, these are the myelin, that's the myelin sheath coating it, right? Individuals that have issues with the myelin sheath, and this is an autoimmune disease, have a condition called multiple sclerosis. And what happens is the immune system is attacking their own myelin around their axons in the central nervous system, and it's causing a lot of problems with signal conduction through these neurons. And it varies widely in terms of the phenotype and in terms of what people look like, both from one person to the other and within a single person over time. So in other words, I have a cousin that has multiple sclerosis. I have two actually. And depending on the day you see them, you may realize they have multiple sclerosis, where their speech is affected a little bit, they slur it a little bit, or their movements are a bit slower than normal, right? Other days you wouldn't even know they had it. So it varies tremendously, from day to day within a given person, and there is a decline over time. But even within people, some people are very severely affected by multiple sclerosis where they're in a wheelchair, and other people don't have that happen for many, many years. So it's very sort of interesting. The cause is not exactly known, but it does affect more women than men. But when men are affected, the disease usually progresses more quickly and it's more devastating. But there's a lot more that we have to learn about this disease. But again, the connection is that it attacks the myelin, right, around the axons. If you want to learn more about that, you can go to the National MS Society, and whenever you want to learn more about any disease, it's always nice to go to their society's website because they explain it in an intelligent, correct, but very understandable way when you're first learning about a new disorder. Okay, so this was our lecture on the nervous system, specifically tissues and organization, and these are our learning objectives here. Before you proceed to the next lecture, make sure that you know these learning objectives, and then next time we'll talk more about different aspects of the nervous system.